Good afternoon, everyone. I hope all of you are doing fine. All right. So, so we had basically learned about the ribosomes in our previous session. So now we'll be learning about the Golgi bodies. How are they related to it? So first, who discovered? Camillo Golgi, 1898, first discovered dense dense reticular structures in the nucleus. They consist of many flat disc-shaped sacs or cisternae, and they were known as the Golgi Apparatus, Golgi bodies, or the Golgi complex. These are stacked parallel to each other. So I'll probably show you the image so that it's more clear. So this is the image of the Golgi apparatus or Golgi bodies. I'll be explaining how it functions also. So the next thing is the varied number of cisternae are present in the Golgi complex. So I hope you can see this marked as cisternae. Yes. Next thing. The Golgi Cisternia concentrically arranged near the nucleus with distinct convexes or the forming phase and concave trans or the maturing phase. The cis and the trans phases, so these are basically the two phases of the organelle, are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packed in the form of vesicles. As I told you, they pack them in the form of vesicles. I hope you can notice this is the vesicle. They are transport vesicle from the Golgi. That this explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. So the things to notice which I'm gonna tell you. So let me just recapitulate that what I had just summarized, what I had told you of formation of proteins. The nucleolus, which is present in the nucleus, produces ribosomes. Now these ribosomes, they help in the synthesis of proteins. So they attach to the surface of endoplasmic reticulum and they form the rough endoplasmic reticulum or the RER. So when they here the synthesis process takes place, and when they are synthesized a bit, they basically are transport vesicle from rough ER. From rough ER, you can see they come and reach to this cis phase. This phase of the Golgi apparatus is the receiving side of the Golgi apparatus. From here, the vesicles they receive. Okay, this basically you can notice. So now they what happens over here? Over here again, this Golgi, it attaches, this Golgi body attaches them with lipids, then with this proteins and all some other type of proteins and acids, many things attached to it. And the actual structure, it's like this, the post office of the cell. Let me post office of the cell. If you just compare it in such a manner, it will be easy for you to understand. Post office of the cell. Here the materials are packaged. Packaging center. So if you go in a biscuit factory, over there the biscuits are packaged, right? So that packaging center is known as the Golgi apparatus over here. Packaging center. Let me mention that. Packaging center. Alright, so that's what they do. And they release in the form of vesicles. You see, these are the vesicles, they are just forming. Secretory vesicle from transphase. Transphase is the shipping side of the Golgi apparatus. From here, the shipping means the removal of transport vesicle from the Golgi. So these vesicles, they come over here with some sort of synthesized proteins, and some I hope some ribosomes also enter. These are basically the, you can see the proteins also. The proteins, okay, when they attach with this Golgi bodies, they 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 are released out of the secreted out of the cell, to give, so that the body functions in a proper manner. And also in the cell, yes, even cell is a living organism. Living, it's living, right? We are not an organism. So this is what the function of Golgi apparatus or Golgi bodies or Golgi complex. They are packaging centers. Packaging centers. So I hope you understood that how the protein synthesis occurs. So majorly we are going to focus on the synthesis of proteins. The next thing is lysosomes. Now what are these lysosomes? These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. Yes, so they are also formed in this Golgi apparatus. So it produces lysosomes. Produces lysosomes. Now, what are the works or what is the work of the lysosomes? These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. They are known as the suicidal banks. So, they are the suicidal banks. Now, I'll consider in same manner the biscuit factory itself. So in that biscuit factory, the suicidal bags, all those broken biscuits, all those waste or which have not been properly made, those are just sent in the suicidal bags. They're just in one area, they just keep it, right? Same way over here, lysosomes. It digests dead cells, also it digests unwanted materials also. Okay, so they are produced, I will just show you the image of this lysosomes. Yes, so this is the image of a lysosome. So it is a membrane bounded structure. It has some enzymes, it transport proteins. So some exocytosol pads of the cell digest waste materials. So I hope you understood. These are the structures of a lysosomes. All right. So this like the waste, the way thrown. Just you can take dustbin, but dustbin is not capable of digesting the waste and releasing out the useful ones, but they are capable. The next is the mitochondria. I would say that it's also very important. It's the energy house, powerhouse of the cell. So this cell carries out so many functions: synthesis of proteins, lipids, carbohydrates, nucleic acids. These many things the cell carries out. So from where does it get the energy? From where does it get this energy? That's also some point to think, right? So it gets energy from the mitochondria, which is the powerhouse of the cell. Each mitochondrion is a double membrane structure with the outer membrane and the inner membrane, dividing its lumen distinctly into two aqueous compartments. This is the outer compartment and the inner compartment. The inner compartment is called the matrix. The outer compartment forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infoldings called the cristae, singles crista, to burst the matrix. The cristae increase the surface area. Mitochondria are the size of aerobic respiration. Now, what do we mean by aerobic and anaerobic respiration? I hope you know what is aerobic and what's anaerobic respiration. Aerobic respiration is respiration that takes place in the presence of oxygen. Respiration that occurs in the presence of oxygen. Right. So this mitochondria, they produce cellular energy in the form of ATP, hence they are called the powerhouses of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecules, ribosomes, 70S, and the components required by the synthesis of proteins. The mitochondria are divided by fission. So this is these points I hope not that important. Just the major thing is this is a site of respiration, site of aerobic respiration, then energy. Here ATP. Do you all know the full form of ATP? Adenosine triphosphate. Adenosine triphosphate. Okay, adenosine triphosphate, which stands for ATP, stands for those. Okay, so here is the structure of your mitochondria. So I had told some infoldings. This is the crustate. Yes, or is yes, the inner membrane forms an infolding called the crustate over the matrix. Now, what is the matrix? This is the matrix. This is a watery kind of substance contains the inner compartment is known as the matrix, the outer membrane, the inner membrane, the crustate, the enfoldings. So this is basically the power house of the cell. So it stores energy in the form of ATP. It provides energy to all the organelles. Provides energy to all the cell organelles. So I hope you understood till here. In our next topic, we'll be learning about vacuoles and all. So today we learned about the mitochondria, the lysosomes, and the Golgi bodies. So I hope that this was clear enough for you all. And if you have any doubts, kindly post it in the comments box. And and shall I give you any question to answer? To think? No, it's not required. In every session, it's not required, right? So the one thing, just revise these things to be as to be more clear with it. And thank you. Keep smiling. Thank you.